Asparagine is one of the 20 most common natural amino acids on Earth. It has carboxamide as the side chain's functional group. It is not an essential amino acid. Its codons are AAU and AAC. A reaction between asparagine and reducing sugars or other source of carbonyls produces acrylamide in food when heated to sufficient temperature. These products occur in baked goods such as French fries, potato chips, and toasted bread. History: asparagine was first isolated in 1806 in a crystalline form by French chemists Louis-Nicolas Vauquelin and Pierre-Jean Robicot from asparagus juice, in which it is abundant, hence the chosen name. It was the first amino acid to be isolated. 3 years later, in 1809, Pierre-Jean Robicot identified a substance from licorice root with properties he qualified as very similar to those of asparagine, and that Plisson identified in 1828 as asparagine itself. Structural function in proteins: since the asparagine side chain can form hydrogen bond interactions with a peptide backbone, asparagine residues are often found near the beginning and the end of alpha helices, and in turn motifs in beta sheets. Its role can be thought as capping the hydrogen bond interactions that would otherwise be satisfied by the polypeptide backbone. Glutamines, with an extra methylene group, have more conformational entropy and thus are less useful in this regard. Asparagine also provides key sites for N-linked glycosylation, modification of the protein chain with the addition of carbohydrate chains. Typically, a carbohydrate tree can solely be added to an asparagine residue if the latter is flanked on the C side by X-serine or X309, where X is any amino acid with the exception of proline. Sources: Dietary sources asparagine is not essential for humans, which means that it can be synthesized from central metabolic pathway intermediates and is not required in the diet. Asparagine is found in animal sources: dairy, whey, beef, poultry, eggs, fish, lactalbumin, seafood, plant sources, asparagus, potatoes, legumes, nuts, seeds, soy, whole grains. Biosynthesis: the precursor to asparagine is oxaloacetate. Oxaloacetate is converted to aspartate using a transaminase enzyme. The enzyme transfers the amino group from glutamate to oxaloacetate, producing I+ or minus ketoglutarate and aspartate. The enzyme asparagine synthetase produces asparagine, AMP, glutamate, and pyrophosphate from aspartate, glutamine, and ATP. In the asparagine synthetase reaction, ATP is used to activate aspartate, forming I2-aspartyl AMP. Glutamine donates an ammonium group, which reacts with I2-aspartyl AMP to form asparagine and free AMP. Degradation: asparagine usually enters the citric acid cycle in humans as malate. In bacteria, the degradation of asparagine leads to the production of oxaloacetate, which is the molecule which combines with citrate in the citric acid cycle. Asparagine is hydrolyzed to aspartate by asparaginase. Aspartate then undergoes transamination to form glutamate and oxaloacetate from alpha-ketoglutarate. Function: asparagine is required for development and function of the brain. It also plays an important role in the synthesis of ammonia. The addition of N-acetylglucosamine to asparagine is performed by oligosaccharyltransferase enzymes in the endoplasmic reticulum. This glycosylation is important both for protein structure and protein function. Betaine structure. External links: GMD spectrum, Why Asparagus Makes Your Pee Stink. References.